Now, I would like to welcome Professor Kai Toshie from the Graduate School of Frontier Biosciences. The lecture title is Fruit Flies Tell the Secret of Germ-line Cells. Small RNA Safeguard the germ-line genomes from the attack by transposons. Professor Kai, please.
Yes. Hello, everyone. My name is Toshie Kai, and I'm a full professor in the Graduate School of Frontier Biosciences, Osaka University. Today, I'm talking about my favorite fruit flies. So the title is Fruit Flies Tell the Secret of Germ-line Cells. You might think, well, fruit flies are just insects, but fruit flies have tremendously contributed to the life science, advances of life science, and also for the good science education. So, and I've been fascinated with germ-line cells and small RNAs, which safeguard the germ-line genomes from the attack by transposons. So what is the small RNAs and what is transposons? Let's learn about it. Before that, let me introduce myself briefly. So I got a PhD in Japan, actually here in Osaka University, without knowing I would be coming back as a faculty. And then I went to the States as a postdoc in the Carnegie Institution, Washington, which is affiliated with Johns Hopkins University. And I became a principal investigator in Singapore and eventually became an associate professor in the National University of Singapore. And I became a food prof here in Osaka University in 2015. Okay, and so I'm interested in germ-line cells. Why? Well, so the individuals, of course, they're dying. You know, the somatic cells, we have a whole bunch of different somatic cells, like including hair, scans, and a bunch of different organs, and they eventually die, and the individual will die. But we can generate next generation by making the eggs, sperms, the gametes, and then they fertilize them together and then give rise to the embryo. And the embryogenesis takes place to develop each individuals. And in each individual, again, we make eggs and sperm. So germ-line cells, which differentiate into eggs and sperms, they are kind of immortal because they make next generations that will be inherited to the next generation. Okay, so the germ-line cells are the drivers. Individual, just a vehicle. So germ-line cells will, discard the individual, and then, but they are driving the species. So as a point of view of the maintenance of the species, germ-line cells are most important cell line in the individual. Okay, and animals do this job. So what is the animal? From including the human beings to the lowest animal, polyphylar sponges. So believe it or not, of course they're animals because they're multicellular eukaryotes that can maintain species by sexual reproduction. Okay, so the sponges or even like the jellyfish or whatever, so they can make eggs and sperms, and then they fertilize together and then make next generations. They're animals. Okay, and so what is the sexual reproduction versus asexual reproduction? So asexual, so let me talk about this one, Hydra, which belongs to the Nidaria in the same group of the sea anemone. I hope you know that. So the Hydra make actually polyp. The small individual is kind of budding out from the body, and it's getting bigger and bigger, will be separated as a totally different individual. So this is asexual reproduction. Genetically, they're totally same. It's clone. However, they can do the sexual reproduction. They can make eggs and then popping out from the body, and then will be released. And then sperms also can be released from the other individual, and then they meet together, fertilize, and then make next generation. Those are sexual reproduction. Let's talk about the Drosophila or ourselves. So we don't have asexual reproduction, but we have sexual reproduction. So this is Drosophila melanogaster fruit fly, somewhere in the room, maybe in the kitchen, and then female, males, they mate together, and then embryo, so fertilized eggs, the embryogenesis takes place and then the first instar larva will be hatching, and then they move into the second instar and the third instar larva, and then eventually become pupa, and then imago, adult, will be coming out. It takes only 10 days, believe it or not, from the eggs to the sexually matured animal. So Drosophila is an arthropod, an insect, but they do the same thing like we do. They make eggs and the sperms, and then those are haploid cells, and then they fertilize them together, and then give rise to the diploid individuals, and then eggs and sperm can be generated again in each individual. So this cycles again and again and again. And so you may wonder why Drosophila. So Drosophila has a long history as model organisms. As you might know, the Thomas Hanborger, he was also called as the father of genetics. So he started around the end of the 19th century, and he started to use the Drosophila as a model organism to study genetics. So and actually Drosophila, wild-type Drosophila has a red eye, but he found out white eyes among the many fruit flies, and then realized that actually child of the mother can have white eyes. So that whatever it is, the something which actually determines the phenotype or nature can be inherited to the next generation. So let's call that one as gene. And he actually found out whatever it is, the gene is on the chromosome. So he was awarded Nobel Prize for the finding, without knowing actually the gene is composed of the DNA. I'm going to talk about in a minute. And then Drosophila, the genomes has been fully sequenced around 2000, and it's been published. And it's a very simple. So Drosophila has only 8 chromosomes, 4 chromosomes times 2. And on the other hand, human beings has 46, right? 23 times 2. So it's very complicated. You cannot do the genetics, but we can do with Drosophila. But believe it or not, for example, the conservation in terms of the genes are very high, even between human and Drosophila. So the genes, which is in charge of the responsible for the genetic diseases, are quite well conserved in Drosophila. So we can even mimic, for example, human genomes like Alzheimer, and you can study about the mechanism with Drosophila about the Alzheimer. So Drosophila is very useful. And so many Nobel Prizes have been awarded to the study using Drosophila. And then this is the very first, the father of genetics, Thomas Hanborger, and his student, Mahler, and many other researchers, including the very last one, like 2017, with the finding of the circadian rhythm in Drosophila, believe it or not. And the first one was even before the finding of or realization of the genes, the DNA as a gene. And as you might know, we all know the deoxyribonucleic acid is composed DNA. But without knowing that much earlier, he proposed that gene determines the phenotype or nature of the individual that can be inherited to the next generation. So that is genetics. So the chromosomes are inside the nucleus in the cell, and in the chromosomes, the DNA is compacted. And then, as you might know, the genetic DNA codes for the genetic information. That's sequence information of the deoxynucleic acid. So from the DNA, the gene is on the DNA, genes are located. And then from the genes, the messenger RNAs are transcribed by RNA polymerases, and then proteins translated from the messenger RNAs. However, there are some RNAs which cannot be translated into proteins, but can function as an RNA This is, for example, tRNA, transfer RNA, and then they can function as RNA. So the genes are not only for the protein coding, but also they can function as non-coding. Okay. And this is the species, a bunch of different species of the non-coding RNA. And as I said, so this is the ribosomes, which is actually translating the messenger RNA into peptide by bringing the tRNAs here. So this is the non-coding, for example, one example of the non-coding RNA, tRNAs, and also ribosomes contains a whole bunch of different ribosomal RNAs. Okay, but not only that, nowadays we know so many different long non-coding, we call link RNA, that can be function as a scaffold of the proteins, or can be function as a precursor for the small RNAs that can actually regulate gene expression. So, and let's talk about the small RNA later, a little bit later. But before that, so the genomes, for example, this is the pie chart of the human genomes. So believe it or not, less than 2% are coding the proteins, and ribosomal RNAs or tRNAs, transfer RNAs, or the rest. And 40% of the human genomes are actually even not our own gene, but derived from the very ancient viruses or just repetitive sequence. We don't know. So they are kind of genetic parasites. And it's kind of shocking to know that our genes is actually parasited with so much different of such genetic parasites. And transposon elements or transposons are actually genetic mobile elements. That's very bad because they can disrupt our genes, which is very important. So there are two kinds of transposon. One is very simple, we call DNA transposon, that can be excised from the original place, original regions of our DNA, and then this DNA intermediate, the excised one, can be simply inserted to the other places. So it's cut and paste, very simple. However, the other one, retrotransposon, derived from the RNA viruses, for example, so they can be transcribed by host RNA polymerase and then can be made into the RNA intermediate. So this is the transcription, so that can be amplified a lot. So you can get lots of RNA intermediates that can be converted to the DNA intermediate by the reverse transcriptase encoded by themselves, and then this DNA intermediate can be inserted to the other places. So because of this amplification cycle, this can propagate a lot. That's very bad. So both cases, they can actually insert to the different places of the genome that could cause the insertional mutations or deletions or whatever. That's very bad, especially in the germ-line cells, because the genomes in the germ-line cells are inherited to the next generation. We have to fight with them. So how? Oh, well, this is the pie chart of how bad we have the genetic parasite. We have 40% of the flies and Drosophila, 40% of the human beings, and 20% roughly in Drosophila. So we have to fight with them. Actually, good. So we have small RNAs which can combat with such a transposons. We call pi RNA. We call pi RNA, but we have other small RNAs which is only like 21 to 29 nucleotides, very short. MicroRNAs, small siRNAs, and RNAs. They have other functions, but let me focus on the RNA, which is of my interest. And the RNAs are being made by the made and bound by the PV family proteins, and the other small RNAs are also bound by the orthogonal family proteins. They are in the same group. So, and then this PV family proteins and RNA complex will repress the translation of the transposon messenger RNA, or will degrade the messenger RNA of transposons. By doing so, they can repress the activity of the transposons. to avoid the confusion or mutation of the genes by the transposon. So this is the structure of the protein of the argonal family or PB family proteins. They have similar structure anyways. And here you have the small RNAs binding here and then target the messenger RNA of the transposons by complementarity in between them. So I'm working on the Drosophila in order to study how the parents are being made. So Drosophila female, if you suck up the abdomen, I will show the movies in a minute. So ovaries are popping out. So there are one pair of ovaries, and in each of them, the small unit called ovarioles are actually bundled into ovary. And in each ovaries, there are developing eggs. We call egg chambers becoming bigger and bigger. Eventually, becomes oocyte or eggs, and it will be laid. Okay, and each germ-line cells, you can appreciate the small punctal granules around the nuclear membrane, and then this is the blown up picture. You can look at the nuclear membrane, and then something is sitting on the nuclear membrane, electrodense structure, and then we call nuage. It's a French word meaning cloud in the sky, because it is amorphous structure, no structure, just membraneless. And here, a whole bunch of different proteins are gathering to process the RNA. This is what I'm studying actually right now. So on the nuclear membrane, there is a nuage, and then there are two different PB family proteins binds, and then chopping up the transposons RNAs or precursors of RNAs, and then this cycle goes around to make a mature RNAs, and replacing the transposons. And now I reported some of the proteins which functions for this small RNA production for the first time in the world. And then I think that's it. This is, I hope everybody can get some idea about what the small RNA is and what the nuage is, and then the importance about the germ-line cells. And let me show you the lab tour. Okay. So this is the main lab. I have lots of PCR machines here, and sorry for the messy decks. And we have real-time PCR and incubators and stuff like that. And we have, of course, deep freezers, liquid nitrogen tank for the cell lines and chemical foods and refrigerators, incubators. Now we are getting onto the fold way, and let's go getting into the fry room. We have double doors because we are working on the genetically modified animals in order to avoid escaping them. So we have double doors. As the one international student from China, we have a whole bunch of different Drosophilas here. Okay, this is my PhD student. She's Japanese. And let's dissect ovaries from flies. So we have CO2 tank. We can anesthetize the flies by CO2. The CO2 is coming to this pad, and you can spread the Drosophila on the pad. So let's dissect. Let's pour the buffer. I think it's just PBS, phosphate buffered cell line, and then you grab the flies. Actually, we are sacrificing the flies every day. Okay, dump into the buffer. So you adjust the zooms and focus. So how it looks like under the dissecting microscope. You will see in a minute. So you grab the legs and then open up the abdomen, and well-fattened ovary is coming out. It's popping up. the intestine, the gut is coming up together. Oh, the one eggs popping here. Okay, we don't need intestine because we're interested in only in oogenesis. So anterior is on the top, and you see the white stuff, that's yolk. The Drosophila yolk is white color, and then the anterior part is translucent. It's very beautiful, isn't it? And they are connected by the oviduct. So if you go to the under fluorescent microscope, actually we dissected the genetically modified Drosophila expressing the enhanced GFP tagged BASA. That's the component of the pyranid pathway sitting on the notch. So you can appreciate the puncta around the nuclear membrane and believe that in each puncta aggregates, the small RNAs are being processed. And this is taken by the confocal microscope by making the optical sections. Variable move is 1 hour, actually shortened into the 10 seconds. I hope you enjoyed. And then in the end, so because I'm a researcher in the biomedical field, I want to introduce them where you can study biomedical research in Osaka University. We have 5 undergraduate schools, School of Science, School of Human Sciences, Faculty of Medicine, School of Dentistry, School of Pharmaceutical Science. And graduate school, we have one more, Graduate School of Frontier Biosciences. This is where I belong. So I highly recommend to consider about this one once you get a master's degree in your home country or somewhere. And actually, I think the FBS, our Frontier Biosciences, is a very internationalized department because the international student rate is 25%. And please check out our curriculum on the website. And thank you so much for listening.
Thank you very much, Professor Kai. The images were very interesting.